Today we'll study the structure of a neuron and transmission of nerve impulse. Neurons are the structural and functional units of nervous system. These are structural and functional units of nervous system. A neuron basically consists of three parts: cell body, which is also called cyton, dendrites, and axon. Cell body or cyton is also called perikaryon or soma. Right? If you see the diagram of a neuron in its simplest form, a neuron is like this. These are the dendrites. This is axon. Axon is the longest part of a neuron. These are terminal endings of axon. This is nucleus of neuron. These are dendrites. This is axon. In the cell body of neuron, small granule-like structures called Nissls granules are present. Nissls granules are ribonucleoproteins which are concerned with synthesis of enzymes required for the formation of neurotransmitters. Nucleus of neuron is suitable for Barr body staining. Barr body is the inactive X chromosome of the female. Plasma membrane of axon is called axolemma. Inside the axon, exoplasm is present. A neuron may be covered by myelin sheath, which acts as an insulator. The cells which are producing myelin sheath, they are called Schwann cells. So this is a Schwann cell. Schwann cell produces myelin sheath on its inner side. So here we are showing myelin sheath. There are certain areas in the axon where myelin sheath is absent. Those gaps are called nodes of Ranvier. So at the node of Ranvier, myelin sheath is absent and even Schwann cells are absent. All the Schwann cells taken together, they form a sheath called neurolemma. Right? So the outermost layer will be neurolemma, which is the sheath of Schwann cells. Below neurolemma, we have myelin sheath, which acts as an insulator. Those areas where myelin sheath is absent, they are called nodes of Ranvier. Right? So this is the basic structure of a neuron. This neuron is having myelin sheath, so it is called myelinated neuron. Other name is medullated neuron. All the neurons are not myelinated. There are certain neurons in our body which do not have myelin sheath. They are called unmyelinated neurons or unmedullated neurons. Now, the basic function of a neuron is to conduct nerve impulse. Now, we'll see how a neuron conducts nerve impulse. This is axon. This, as we have discussed, is the plasma membrane of axon or axolemma. Outside axolemma, we have ECF, that is extracellular fluid, sodium ions, potassium ions, chloride ions, and bicarbonate ions. They are present in ECF as well as inside the exoplasm. if we see enlarged view of exolemma, we find that it is a bilayer of lipids. These are lipid molecules with hydrophilic and hydrophobic ends. This lipid bilayer is an insulator. Lipids are not going to allow the exchange of these ions. Exchange of ions will occur through specific channels. So here I have drawn a channel. This channel is actually a transmembrane protein. It is present in the lipid bilayer and it is a transmembrane protein. If sodium ions have to move from ECF to exoplasm, these can move only through these specific channels. Similarly, if potassium ions have to move from inside to outside, they move through these channels. So here we can see that there is influx of sodium from ECF, that is extracellular fluid, to exoplasm, and here we can see there is efflux or exit of potassium from exoplasm to extracellular fluid. Now, the three stages of impulse transmission are: first, there is polarized state, when the neuron is polarized. Second stage is depolarized state, and third stage is repolarized state. Step by step, we'll see all the three stages of impulse transmission. First, let's see the polarized state of a neuron. This is axon. Inside the axon, we have exoplasm. Outside, it is ECF. Now, during normal polarized state, inside of the exolemma is negatively charged, and outside of exolemma is positively charged. This diagram is of polarized state. It is also called the normal resting stage or the state of neuron. Outside positive, inside negative. If you connect sensitive microelectrodes, we find there is a transmembrane potential of -70 millivolts. This is called resting membrane potential or transmembrane potential. So the value of RMP or resting membrane potential is -70 millivolts. Now the question comes, why it is negative and why it is positive outside? If we say that it's the play of sodium and potassium ions, then both are positively charged. How is it possible that inside we have negative charge and outside we have positive charge? The reason is that if we compare these two channels, the number of potassium leakage channels, the channels which are allowing the exit of potassium, that is more in exolemma as compared to number of sodium leakage channels. So naturally, more number of positive ions is leaving the exoplasm as compared to the number of positive ions moving inside. So more of potassium is moving out and less of sodium is moving in. Plus, in the exolemma, we have a structure called sodium-potassium pump, which keeps on throwing 3 sodium ions out and it brings 2 potassium ions in. Means more of the positive charge is leaving the cell or the exoplasm and less of positive charges coming inside. This sodium-potassium pump uses 1 ATP. One question still remains, that both the charges are positive, then why we are saying that inside it is negative? That means we must have some negatively charged ion also here, and those negatively charged ions are immobile protein ions. I'll repeat, immobile protein ions and phosphate ions are more inside the exoplasm. Now, because they are immobile, they cannot leave the exoplasm and they cannot move out. So the net result becomes that inside we have negative charge and outside we have positive charge. So this is the normal polarized state of neuron, in which the resting membrane potential is minus 70 millivolts. The second stage of impulse transmission is depolarized state. Now let's see how depolarization occurs, or how the change in polarity is brought about. This is axon. In normal state we said inside is negative, outside is positive. When we give a stimulus to this axon, stimulus is any kind of change in the external environment. This stimulus can be mechanical, can be chemical, can be physical. The stimulus has to be of threshold value. Threshold means minimum value of the stimulus which is capable of generating a response. If we apply a threshold stimulus, then membrane polarity reverses in a short patch of axolemma. That means inside becomes positively charged and outside becomes negatively charged. The polarity reverses.